We have reached unit number 5 in the AP Biology curriculum. This is one of my favorite topics, heredity. It's the study of how traits, characteristics are passed down from parents to their offspring. And I think it's one of the most interesting parts of biology. It's certainly one of my favorite things to teach. We're going to cover pages 214 to 221 in the Biology in Focus textbook. I don't know that I'll cover all of that in this one recording. I will likely break this up into multiple sections, but this is kind of roughly where we are in the textbook. And the title of today's lesson is Mendel and the Gene Idea. So we're talking about inheritance, and not the kind of inheritance where someone dies and they pass down their money and their wealth to the next generation, but inheritance as in the passing down of traits, characteristics from parents to offspring. And as long as there has been a human race on the earth, humans have recognized that this occurs, that somehow children look like their parents. And you can look in the mirror and you can probably see physical traits, maybe the shape of your nose or the shape of your ears or your hair color, that clearly seem to be linked to your mother's side of the family. And you might see other physical traits that seem to have been passed down from your father and his side of the family. And so humans have always known that somehow this happens. There are lots of references in ancient writings. You can, even in the Old Testament of the Bible, there are phrases that reveal that the ancient Hebrews understood very well that by breeding sheep, for example, with different kinds of characteristics, different color wool, or whatever the characteristics may be, that somehow you could sort of control how the offspring would turn out by carefully choosing which parents you bred together in the livestock. So humans have known for a long, long time that somehow parents pass down characteristics to their offspring. The question is, how? How does this work? And for a long, long time, nobody knew. But the scientific study of inheritance is genetics. Genetics is a relatively new branch of biology. It has only been recognized as an official branch of biology for about 120 years. It was right around 1900 that the work of Gregor Mendel was rediscovered, and it's kind of a sad story. Gregor Mendel lived in the 1800s. He did his research in the 1850s and 1860s. Lots of experimentation, wrote up a wonderful paper explaining how genetics works, explaining the basic rules and laws of heredity. But he was so far ahead of his time that nobody got it. Nobody understood it. The other scientists of the day just did not understand what Mendel was saying. And so his ideas did not catch on. And unfortunately, it was not until about 16 years after Gregor Mendel's death, about 34 years after his research was published, that finally he was recognized for what he had accomplished. And it was that rediscovery of Gregor Mendel's research, again, 16 years after his death in the year 1900, right at the beginning of the 20th century, that's when the branch of genetics first shows up. Now, prior to the work of Gregor Mendel, nobody understood how traits are passed down from parents. But there were three different competing hypotheses that emerged. One was called blending hypothesis, also known as blended inheritance. And this basically says that the traits or the genetic information that's passed down from your mom and your dad, that they kind of blend like paint, almost like if you took black paint and white paint and mixed it together, you get gray. Or if you take blue paint and yellow paint and blend it together, you'll get green. So the idea was that if a tall father marries a really short mother, that all of their children will be medium height. Or if a black squirrel mates with a white squirrel, you're going to get a gray squirrel. And you can see why this hypothesis was popular, because it seems to be legitimate, at least in some situations. There are some problems with it, though. If the blended inheritance model were correct, and if a tall man produces children with a short woman and you get medium-sized children, okay, maybe that happens sometimes. But then that would mean that those medium-sized kids, when they grow up and produce children, they could only pass down medium-sized genes. The tall gene and the short gene would no longer exist because they've been blended together to make a new gene, a medium-sized gene. Of course, it doesn't work that way. We know today that this hypothesis is not correct, although sometimes it seems to be. The incubation hypothesis was another idea that we now know to not be correct, but the idea was that only one parent controlled the characteristics or traits of the children. And this goes back to the 16, 17, 1800s, which was a very male-dominated society, so most people were spermist, meaning that they thought that inside the sperm cell was a very, very, very tiny preformed embryo already inside the sperm cell, and that all of the physical characteristics were passed down from the father, and that the purpose of the egg was just to provide nourishment for the growing embryo. Of course, there was another group called ovists who thought exactly the opposite. They thought that inside the egg was a very, very tiny preformed child, and that the sperm's role was just to trigger the development of that tiny, tiny embryo, but that all the physical characteristics came from the mom. And both of those ideas are completely ridiculous. It's pretty easy to dismiss the incubation hypothesis. The one that turns out to be correct is the particulate model. And I'm going to show you how we know it to be correct. But the particulate model says that when parents pass down their traits to their children, that what they are actually passing down are discrete units, particles, that retain their identities. They don't blend together. Black does not blend with white to make gray. Tall does not blend with short to make medium. In other words, genes are particles and they do not blend. And we know this to be correct because Gregor Mendel proved the particulate model to be the correct model. And we're going to learn exactly how he did this very shortly. All right, so here he is, my hero, Gregor Mendel. Mendel's story is a fascinating story, and his accomplishments are truly extraordinary for a lot of reasons. This man lived most of his adult life in a small monastery. He was a monk in the Catholic Church, and lived in a small town that is now part of the Czech Republic, but back then was part of Austria. And Mendel's accomplishments are extraordinary, not only because he was a monk from a very poor farming family, not just because he only had 2 years of college training, and the only reason he had any college training was because the church paid for his education once he became a monk. But the main reason why I say his accomplishments are extraordinary is Mendel was one of the first, maybe the first, scientist to use statistical analysis, mathematical analysis of data to solve a biological problem. Now you might think, well, wait a minute, math is a common part of science. Why is that so extraordinary? Well, in the 1800s, physicists used math in their calculations, and chemists used mathematics, but biology was not a real mathematical science in those days. The idea of using statistical analysis of data, number crunching, to solve a biological problem, this was a fundamentally new concept, and one that was pioneered by Gregor Mendel. And because of that, because he based his findings on statistical analysis, he was so far ahead of his time that, as I mentioned, the other scientists of this time period just didn't get it. They didn't understand what Mendel was trying to tell them. And what he was trying to tell them was that the basic rules and laws that govern how inheritance works follow very predictable patterns. And he was able to figure out 4 basic concepts that explain essentially how genes work. And so we're going to learn how Mendel did this. In 1856, he began a long series of breeding experiments to figure out how traits are passed down from parents to offspring. He began his research with rodents, crossing, black mice with white mice, or crossing long-tailed mice with short-tail parents, because he wanted to see how the traits would be passed down from one generation to the next. Well, the animal breeding was kind of frowned upon by the church, and so Mendel had to sort of put a halt to that research, and he switched over to plants, specifically pea plants, which could easily be grown and cultivated right beside the monastery where he lived, in the garden. One of his jobs in the monastery was to tend the garden because the monks grew their own vegetables. And he thought, wow, I can kill 2 birds with 1 stone. I can grow delicious vegetables to feed all of my friends here in the monastery, but I can also conduct my research in the garden with pea plants. Now you might think, why in the world would he choose pea plants? Peas make an excellent choice for genetics research because they have a short lifespan. It only takes a few months for a pea plant to reach maturity. Pea plants are monoecious, which is a fancy way of saying that both male reproductive and female reproductive parts are present on the same plant. In fact, they're on the same flower. Every flower, every bloom on a pea plant has both male and female parts. Pea plants are capable of cross-pollinating or self-pollinating. I'll explain that in a moment. But one reason why Mendel chose peas is because there's lots of different traits. You don't think of pea plants as being all that exciting, but there are 7 different physical characteristics that Mendel identified, and they all turn out to be genetically inherited. Not to mention, another great reason to use pea plants is after you're finished with your research, you can eat the peas, right? So here is a short summary of the 7 different characteristics that Mendel studied. Flower color. Some pea plants have purple flowers, some have white. Flower position. Some pea plants form their blooms branching off of the main axis of the plant. Other pea plants produce all of their blooms at the terminus or the end of the stem. Seed color, some pea plants have yellow seeds, some have green. Some pea plants have round seeds, some have wrinkled. Some pea plants have smooth pods and some have wrinkled pods, green pods or yellow pods. And finally, some pea plants are very tall, about 6 feet tall, and other pea plants are much, much shorter, only about 2 feet. And all of these characteristics are determined by genes which are passed down from pea plant parents to their offspring. And so Mendel wisely realized that if he could understand how this works in pea plants, if he could uncover the basic rules and laws that explain how pea plant genes work, those same rules probably apply to other organisms, even humans. And for the most part, that turns out to be true. So Mendel began his research with cross-pollination experiments. And so he would take 2 separate pea plants, those were his parents. He would transfer pollen from the male part of one plant and transfer it over to the female part of a different plant. And then the babies, the offspring, were called hybrid offspring, and he would simply let them grow to see how they looked, to see what kind of traits were passed down from the parents. So here's a close look at how this works. So you can see in this case, Mendel was crossing a white-flowered pea plant with a purple-flowered pea plant. Both of these parents are what we would call homozygous. That is, the purple plant only has genes for purple and the white plant only has genes for white. And he would take a little paintbrush and he picked up some pollen from the male part called the stamens, and he would transfer that pollen over to the stigma, which is part of this female part here is called the carpel, and the little tip is the stigma. And basically when you put pollen on the stigma, sperm cells travel down this tube and they fertilize the eggs that are forming inside this little area here. So this is cross-pollination. Now notice Mendel used scissors to snip, snip, snip the male parts off of the purple flower because he wanted to make sure that the only pollen that was allowed to fertilize these eggs on this purple plant was the pollen that came from the white plant, the white flowered plant. All right, the next step after he transferred the pollen was to wait a few months until this little part here, the carpel, matures to form a seed pod. And of course, inside there's going to be the seeds, which are the peas, right? So these are the baby plants. These are the offspring produced by these 2 parents. This is like a daddy, a mama. These are the babies, right? And so Mendel would harvest the peas. He would let them dry out. And then the next growing season, he would plant those seeds in the ground, let them grow. And this was called the F1 generation, or the first generation of baby plants. And he would simply look to see what fraction of the offspring had purple flowers. And turns out, in this case, they all have purple flowers. And we'll talk about why that is. Okay, so here's a closer look. The original 2 parents that were cross-pollinated they're called the P generation, capital P, that stands for parents. And so this was a true breeding purple flowered plant. True breeding just means that this plant has purple flowers, its parents had purple flowers, their parents had purple flowers, on and on and on. This is a purple flowered pea plant that comes from a long line of nothing but purple flowered pea plants. And when we say this is true breeding white, It's a white flowered pea plant. Its parents had white flowers. Their parents had white flowers. You get the idea. Mendel cross-pollinated. Remember, he transferred pollen from the white plant over to the purple plant, allowed the seeds to form, planted the seeds, and then in the next generation, which is called the F1 generation, the first generation of offspring, notice all of the offspring turned out purple. Well, that proves right there that the blended inheritance hypothesis is wrong. Because if purple and white were blending together, then these plants would have light purple color. They would have pale purple, whitish purple. But they don't. They're the same color purple as this parent. But it does bring up an interesting question. What about the white? How come none of these offspring are white like their father here? They all look like their mama. How could that be? Well, then it gets more interesting. Because what Mendel did next was he allowed these purple plants, these hybrid purple plants, to self-pollinate. And in the next generation, which is called the F2 generation, he got both purple and white flowered offspring. And you can see some of the numbers. These are numbers from Mendel's actual research that was published in 1866. But Mendel noticed in his numbers that there's a pattern here. 705 purple flowered plants, 224 white flowered plants. That works out to be very, very, very close to 3/4 purple, and only 1/4 of the offspring are showing the white trait. Mendel figured that must be significant. There's got to be a reason why it always turns out this 3-to-1 ratio. And he also thought it was interesting that even though the white color completely disappeared in the F1 generation, it shows back up in the F2 generation. Even though we don't see any evidence of white genes, we know they exist because all of a sudden that trait, that white flowered trait, does show back up in the F2 generation. Now, to better understand what's happening here, we can look at a Punnett square, which is a useful way to visualize what's going on with the genes. Gregor Mendel was the first to be able to say definitively that genes are like particles, and that every pea plant has not one, but 2 genes for flower color. In this case, both genes were coding for purple flowers, big P, big P. While this plant has 2 copies of the white allele, which we show as a little p, lowercase p. So if you show that cross on a Punnett square, that means this plant can produce 2 possible types of sperm, and this plant can produce, well, really I should do that the other way around, because we said this was the father plant. So let's say 2 possible kinds of sperm, right here they are, little p, and then this would be 2 possible kinds of eggs from the mama, big P, big P. And so basically what you do on a Punnett square is you're showing what happens if this sperm fertilizes this egg. Well, you're going to get a big P and a little p, right? If this sperm fertilizes this egg, you get this offspring. You get the idea. This sperm, this egg, boom, you get this offspring. So the Punnett square shows us the 4 possible offspring that could be produced from these 2 parents. Okay. Well, you can tell from the Punnett square, there's really only one possible outcome. Every single offspring is going to get a big P from this parent and a little p from this parent. That means this plant has a purple allele and a white allele, but only the purple allele is expressed. Mendel called this dominance. even though this plant has a white gene, it does not show the white color. Purple apparently is dominant and white is recessive. That's why we show white as a lowercase letter, and we show the dominant allele for purple as a capital letter. This brings up the extra credit phrase that pays for today, and it's got a little bit of alliteration. Here it is. Purple flowered peas proved the particulate model perfectly. That's hard to say. Purple flowered peas proved the particulate model perfectly. You know the rules. First 10 to email me that phrase get some extra credit. Okay, so whenever you have a parent that has two genes that are the same allele, the same form of the gene, like big P, or little p, we refer to that as homozygous. Homozygous. Homo means same. Same two genes. Same two genes. But in an offspring like this, where the two genes are different alleles, a big P and a little p, we call that heterozygous. Hetero meaning different, two different alleles. Okay, so the next step, remember, is to allow this heterozygous hybrid plant to self-pollinate. Pea plants can do that. They naturally, very often, will self-pollinate because remember the flower, the pea plant flower, has both male and female parts. And so you simply allow the plant to self-pollinate. So we're crossing a big P, little p with itself. It's pollinating itself. Here's what it looks like on a Punnett square. And we're going to show all of the possible offspring that could be produced for the F2 generation. And there you go. Now remember, big P is dominant. So any plant that inherits even just one copy of big P is going to have purple flowers. If it gets two copies of big P, purple flowers. If it only gets one copy of big P, purple flowers. The only way a plant can have white flowers is if it inherits two copies of that recessive allele, little p, little p. And Gregor Mendel discovered that in the F2 generation, it's always 3-to-1. 3/4 of the offspring will have purple flowers, and 1/4 of the offspring will have white. Okay, remember, whenever you look at a Punnett square, the genes across the top normally represent the different types of possible sperm that could be produced by the parent. And the letters down the side represent the different types of eggs that could be produced by the other parent. And so the 4 boxes in the middle of the Punnett square, these 4 boxes represent possible offspring. And so, this shows me that there's going to be a 1 out of 4 chance when this plant pollinates itself, there's a 1 out of 4 chance that I'm going to get a big P offspring. There's a 2 out of 4 chance that I'm going to get a heterozygous purple offspring. And then there's a 1 out of 4 chance that I'm going to get a little p white flowered offspring. So if you look at that all together, that means there's a 3 out of 4 chance that I'm going to get purple flowers in my offspring, and only a 1 out of 4 chance of white flowers. And that matches up just perfectly with the actual numbers that Gregor Mendel got. Now, if you do the math, you'll find 705 is not exactly 3/4, and 224 is not exactly 1/4, but it's very, very close. And these ratios that are predicted by the Punnett squares, they're just that. They're predictions. It's not always going to perfectly match the actual numbers that you see in the data. So here's a closer look at Mendel's F2 generation results. Again, it's a 3-to-1 ratio because 3/4 of the offspring had purple flowers, and you can see there's a couple different ways. It could be homozygous purple, or it could be heterozygous purple, and then only 1 out of 4 had white flowers. And of course, the only way a plant can have white flowers is it has to be homozygous, little p, little p. It's important to understand the vocabulary, and you've already heard me throw around some terms here. You want to make sure you're getting a handle on the vocabulary, because if you don't understand what these words mean, you're going to get really lost really quickly. So let's go through some of the terms. You'll hear me talk a lot about phenotype and genotype. Phenotype is the physical appearance of the organism, or the physical expression of the genes. So just remember, pH in the word physical, pH in the word phenotype. So if I was to show you a pea plant, and you look at it and it's covered in purple blooms, obviously its phenotype is purple flowers. Phenotype is how it looks. Genotype means what type of genes did it inherit from its parents. In other words, genotype is the genetic makeup of an organism. So if we're talking about flower color, the possible phenotypes would be purple flowers or white flowers. But if we're talking about genotype, it's either big P, homozygous purple, or big P, heterozygous purple, or little p, which is homozygous white. Okay, genes can have alternate versions of themselves, and we call these alternate versions of a gene alleles. So for example, the gene for flower color in pea plants has 2 alternate alleles, the purple allele and the white allele, or as we often say, big P or little p. Those are the 2 alleles for flower color in pea plants. You've already heard me use the terms homozygous and heterozygous quite a bit. Homozygous means that the 2 genes are both the same. Homo means same, the same allele. So big T for tall, or little t for short, or big P for purple, little p for white. If the 2 genes are both the same, it's homozygous. If the 2 genes are different alleles, like big T, little t, we call that heterozygous. Hetero means different. Homo means same. Heterozygous is when the 2 genes are different alleles. All right, a test cross is another term that you need to be familiar with. A test cross is whenever a scientist breeds an organism, intentionally breeds an organism of unknown genotype with another organism that is homozygous recessive. Now, why would you do this? There's a perfectly good reason. If I was to show you a pea plant, and the only thing I tell you about it is look at it. It's a purple flower pea plant. In other words, I don't tell you any background. I just show you the plant, and it's got purple blooms on it. Well, you're going to know the phenotype. The phenotype is obvious. Purple flowers. That is its physical appearance. That is its phenotype. However, you cannot tell just by looking at a purple flowered pea plant whether its genotype is big P or big P little p. It is impossible to tell just by looking whether a purple flowered pea plant is homozygous or heterozygous. But one way you could figure it out is to take that purple flowered pea plant and cross-pollinate it with a white pea plant, because you know the white pea plant has to be little p, little p. So if you cross a purple pea plant with a white pea plant, and if all of the offspring, every single one of them has purple flowers, well, then you know that this parent must have been big P, big P. But if half of the offspring have purple flowers and half have white flowers, then you know that that purple parent must have been heterozygous, big P, little p. So the purpose of a test cross is to figure out which of these two is the correct genotype for the plant, or whatever. I'm just using plants as an example. Whatever organism you're interested in. Well, this is a good stopping point, a good place for us to end part 1 of Unit 5. Don't forget to email me with the extra credit phrase that pays that I mentioned a little bit earlier. And guys, I hope you have a great day.